Hello my dear friends, my name is Mega and welcome to my channel Score Points. In the previous videos we have learned about the male reproductive unit. In this video we can learn about the female reproductive unit. The pistil or gynecium represents the female reproductive unit and the gynecium is mainly of four types: monocarpellary, multicarpellary, syncarpous and apocarpous. Monocarpellary is the gynecium which has a single pistil. Multicarpellary is the gynecium which has more than two pistils. Syncarpous is the gynecium which has fused pistils. And apocarpous, the gynecium which has free pistils. Now we can see the diagram of a pistil. This is a pistil of a hibiscus. Multicarpellary syncarpous pistil of papaver. And now you can see the multicarpellary apocarpous gynecium of michelia. We can learn the major parts of a flower. Before learning this, please watch the dissection of a flower video. I have given the link above. So please watch that so that you can get a clear idea of this parts. So first it's stigma. Stigma is a sticky surface that receives pollen grain. The main function of stigma is to receive the pollen grain during the pollination. Below the stigma you have the style, which is long, slender, elongated part. The main function of the style is to connect the stigma with ovary, hold the stigma in order to receive pollen grains. It is through the style that the pollen tube grows and reaches the ovule. Third and the most important part, it's the ovary. It's the basal, swollen part of the pistil. Ovary contains ovules and eggs. The main function of ovary is to protect the eggs and it is a site for fertilization. You can see the diagram, the bottom region I have marked in red. Let's learn about the ovarian cavity or locule. Inside the ovary there is a compartment which is called as ovarian cavity or locule. This is separated by a septum. Next important term, it's placenta, the tissue in ovarian cavity from where the ovule or megasporangium arises. This is the placenta. So ovules are attached to the placenta. Now you can see the structure of megasporangium or ovule. Ovule consists of many parts and the first part that you are going to learn is funicle. The ovule is a small structure attached to the placenta by means of stalk called funicle. Then you have the hilum. The body of the ovule fuses with the funicle in the region called as hilum. Hilum represents the junction between ovule and funicle. Integuments. Each ovule has one or two protective envelopes called as integuments. Integuments are also found in seeds. There are outer and inner integuments which helps in protecting the seed. Next is micropyle. Integuments encircles the nucellus except at the tip where there is an opening called as micropyle. On the bottom portion or the basal part of the ovule, opposite to the micropylar end, there is a chalaza. Mass of cells enclosed within the integuments, it's nucellus. The female gametophyte located within the nucellus, it's called an embryo sac. An ovule generally has a single embryo sac formed from a megaspore. So we can learn the structure of megaspore and embryo sac in detail. Now we can see the diagrammatic view of a typical anatropous ovule showing all the parts of ovule. Important process that you have to learn in this chapter, it's megasporogenesis, the process of formation of haploid megaspores from diploid megaspore mother cell. It's called as megasporogenesis. In the micropylar region, the nucellus contains cells with dense cytoplasm and prominent nucleus. It differentiate into a single megaspore mother cells. So MMC or the megaspore mother cell, it's diploid in nature and this MMC will undergo meiosis. MMC or megaspore mother cell divides transversely to form two cells called dyad. Again it will divide transversely to form four haploid cells called megaspore tetrad. Out of these four haploid cells, only one remains functional. The rest three haploid cells will degenerate. So this one functional haploid cell develops to a female gametophyte or embryo sac. MMC undergoes meiosis and now you can see the megaspore mother cell. MMC divides transversely to form two cells stage called as dyad. So you can see the two cells dyad structure. Two cell dyad again divide transversely to form four haploid cells called as megaspore tetrad. Now we can see the diagram of megaspore tetrad. Now we can see all the megasporogenesis process. First, the formation of MMC, then you can see the diet structure and also the 4 haploid cell state structure, that is megaspore tetrad. Only one functional megaspore develops to the female gametophyte or embryo sac. This is called as monosporic development because only one functional megaspore will develop, the rest 3 megaspores will degenerate. Next important process that you have to learn in this chapter is megagametogenesis. The formation of female gametophyte or embryo sac, it's called as megagametogenesis. You can see the diagrammatic representation of mature embryo sac on the board. So we'll learn about the formation of this embryo sac in detail. First, it's megaspore. The first cell of female gametophyte, it's the megaspore, and this megaspore will increase in its size and the nucleus divides mitotically into 2 nuclei and move apart to the opposite poles. Thus, a 2-nucleate embryo sac is formed. So you can see the structure of 2-nucleate embryo sac formed by mitotic division of nucleus. The 2 daughter nuclei, again it will undergo mitotic division and it forms 4 daughter nuclei. Thus, a 4-nucleate embryo sac is formed. You can see the 4-nucleate embryo sac on the board. The 4 daughter nuclei, again it will divide mitotically to form 8-nucleated embryo sac. So the important point here is that nucleus is dividing by mitotic process. Now we can see the 2, 4 and 8 nucleate stage of embryo sac and also the structure of a mature embryo sac. The nucleus divides 3 times. It undergoes mitotic division. The 3rd mitotic division gives rise to 8-nucleate 7-celled embryo sac. The central cell, it contains 2 nuclei which is called as the polar nuclei. The 3 nuclei at the micropyla end, it forms the egg apparatus. In the egg apparatus, the middle region, the middle cell, it's the largest and it's called oosphere or egg or ovum, while other 2 cells adjoining the egg cells are called as synergids. The 3 nuclei at the chelacal end are surrounded by cytoplasm and cellular wall. These are called as antipodal cells. We know that there are 8 nuclei in total. 6 of them are surrounded by a cell wall. 2 of them, which is called as the polar nuclei, it is situated below the egg apparatus in a large central cell. Mitotic divisions are strictly free nuclear. After the 8-nucleate stage, cell walls are laid down, leading to the formation of typical gametophyte, which is female gametophyte or embryo sac. Distribution of cells within embryo sac. First is egg apparatus. 3 cells are grouped together at the micropyla end and this constitute the egg apparatus. Egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have special cellular thickening at the micropyla tip called as filiform apparatus, which plays an important role in guiding the pollen tube into the synergids. 3 cells are at the chelacal end and are called as antipodals. The large central cell has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity is called 8-nucleated 7-celled. Now we can see the plot for the above session. This is the end of part B, sexual reproduction in flowering plants. Part C, I'll be uploading it soon. It's about pollination. Thanks for watching. Please like, share and subscribe.